Okay, thank you to Remase for the opportunity to talk about my research. Today, I will talk about the technique that I use, that is small angle scattering, and we will see an example that is to study the nanostructural behavior of proton exchange membrane. I divided the talk in two parts. At the beginning, we'll introduce the technique for who doesn't know about. I will talk about basic equation and some models that are used to analyze the data. And then I will comment the proper research that is in proton exchange membrane. I give a brief introduction about this material, and then we will discuss some results. So the small angle X-ray scattering is an analytical technique that is used to study material structure at nanometric scale. That means that we can get information about size or size distribution. If we have polydispersed system, then we can analyze, for example, the shape of our sample, interaction between them, correlation distance. Is like a really complete technique. And it has some advantage over other techniques that can be like electron microscopy that also are useful to study this nanometric scale. For example, it rarely needs a sample pretendent to study, only requires a small amount of sample, and we can get a lot of information with this. This is like quite important that allow in situ and time resolved experiment. We can have a really complex sample environment, like a magnetic field, temperature, pressure, rheology, and combine with time results, so we can follow the process of reaction or interaction between samples. That is not destructive sample. You can continue to use this sample or study with another method. Here I have to do the clarification that is under certain condition, because now with the 4th generation synchrotron, some samples like more biological or organical can have like a radiation damage. So you have to control this before measure if you want to use your sample for another experiment. So where does the SWAXS signal come from? We have here a basic setup. We have the incident beam, then we have a monochromator to select the energy. Then we have a collimation system to focus the beam into the sample, or it also can be focused into the detector, depending on our setup or our system, what is more interesting. Here in the detector, we will collect the signal coming from the sample, and the sample scattering density, that is which define our sample, can be the electron density in the case if we are using X-ray, or the scattering length density in the case we are working with sun and neutron, will determine the scattering pattern collected by the detector. So the scatter wave amplitude will be the integral of this scattering density multiplied by a phase, with this Q vector, that is the scattering vector, that depend on the wavelength of the incident beam, also with the angle that is scattered our beam. If we pay attention to this expression, we see that this amplitude of the scattering is the Fourier transform of our scattering density that define our sample. So we can think, okay, if we have this function, it's just apply the Fourier transform to have the scattering density, and then we can get for example, the shape or how the molecules are distributed in our sample. But it's not quite easy, since the detector counts incident particles. That means that we have the intensity of the scattering. Neither the module or the phase to this function is measured. If we do some math with the intensity, we can get our module of the function A, but the phase is not accessible. What that means? If we have this density of the sample, so if we know how is defined our sample, we can do some math and get our intensity. But in the case of the experiment, we have the intensity of the scattering, but we cannot reach by doing math to our density of the scattering because this phase problem. We don't know the phase of the scattering wave. So we need to find another method to analyze the data coming from the SWAXS experiment. How will we do? Okay, first let's do to see how the SWAXS data looks like. If we have a well-ordered system like this one that is a mesoporous silica, the SWAXS data will look like similar to a diffraction pattern, like a well-defined peak. If we have like a monodispersed system, we like this one that is a colloidal suspension of latex nanoparticle, we will have a less defined peak, more broad, with well-defined minima. Then if we have more complex system, like a not well-defined shape sample or particle, just we will have like a curve with a well-defined slope. And then for a random coil of a polymer, we will have also a similar curve, but with different slope. So from this different graph, we can get information and can we start to do our analysis of the data. Let's start with the simplest case, like having a diluted system of monoparticles. We can express our scattering intensity as this expression that is taking into account the contrast between the scattering intensity of our particle with the solution, the volume that is occupied by our sample, and then this function. This function is called form factor that will be related with the geometric of our particle. This is a really simple geometric shape, as can be like a sphere. We can define this function analytically. Here we have the example for a sphere of radius r and this expression. If we plot this one, we will have this function with well-marked, well-defined minima, and the position of this minima will be related with our radius. But what happened in the reality is not so common to have a monodispersed system. We have like a polydispersed system with certain like distribution, but we can still analyze the data. Here I plot a Gaussian polydispersed system with average of radius 20 nanometer, and this is the sigma value. We can see that the minima is not well defined, it starts to lose definition, and this broad peak starts to become more broad, but we can still to have the size distribution of our sample. What happens now if we have a not well-defined shape? We can use like a standard plot that are approximation that are useful to extract or infer some parameters. Here I just use two of the more common, that is the linear plot, that is to analyze the data at a low Q value where the particle size is the range of the 2p over q. Here, this form factor is approximated by this exponential expression that is related with this parameter, that is the radius of relation, that give us information on how the density of the scattering is distributed along our sample. And this parameter is related with some characteristic in our sample. For example, here is an sphere. We can relate the radius of radiation with the radius of the sphere. Or if we have a rod, we can get the length of the rod. If we move to a high Q values, we can use the Porot plot, where the particle size is like bigger than this 2p over q. Here, the intensity of the scattering follow a power law function, and this exponent we can use to have information about the dimension in our sample. For example, if it's 1, we can know that it's 1D object. If it's 2, it's 2D object. 4, 3D object. And if we have values between this one, 1 and 2 and 3 and 4, the dimension is fractals. If it's between 1 and 3, we will have like a mass fractal structure. And if it's between 3 and 4, we will have surface fractal. And now what happens if we don't have a diluted system? We have like a concentrate system or a solid. So the particles start interact between them. And now the intensity of the scattering will be related not only with the shape of our object, will be related also with the interaction between particles. So we introduce also this function that is called the structure factor. This function will depend on the kind of interaction that we have between particles in our sample. This is the case where we have like an aggregate system. It's this kind of curve. Then here is like a concentrate system, but the model is hard sphere. So the particles cannot interact between others, just touch and have keep it their space. Or here, that is a well-ordered system, we will have like a well-defined peak. So that is the basic of the technique I will talk about. But I want to keep you in mind that this technique is possible to combine with temperature, like high temperature, low temperature. With pressure, we can stretch the sample and see how the structure change. For proteins, we can use a flow through, also work with the electric and magnetic field. We can control the humidity. Now we are developing like a rheology experiment that you can do like in situ experiment, the rheology with all the complexity of rheology technique has. We have a scanning SWAG that you can have information of the structure in a specific point in your sample with a resolution of a few microns. Then you can do like a SWAXS tensor tomography, or you can do like complex experiment like combining micro tomography with the scanning SWAG imaging. It combined like hierarchical structure in your sample. The good news is all these setup are available at ForMAX via remake. For sure, you can access to another in situ capabilities with another beamline that are also available at remake facilities. So now let's talk about my research. I'm working with the proton exchange membrane. that they are using in fuel cells, that they are electrochemical device that convert the energy of a chemical reaction into electrical energy in an environment-friendly way. I have here like a schematic setup. We introduce the fuel into the anode. Here we have a reaction where it's a chemical reaction that produces proton and electron. The proton will go through the electrolyte to the cathode, and the electron will pass by an external circuit. So this current that is generated here is that we will use. And then in the cathode, together with the oxygen, is another chemical reaction with this product. For the fuel, usually it's like hydrogen, but also can be used some alcohol like a methanol or ethanol. And the membrane that we are studying is a membrane that is used as electrolyte. What we are looking into the membrane. So we need this membrane has high proton conductivity because we want the proton go through the membrane, low electron conductivity because we want to use this electron to generate a current here. Low fuel permeability because if there is crossover of the fuel, we will reduce the performance of the fuel cells. And then the membrane have to resist the operation condition. So it needs to be like high chemical, mechanical, and thermal stability. So the motivation for this study are basically two. First one, we wanted to test a new setup that is a combination between the dynamic mechanical analysis with the small and wide angle X-ray scattering, the possibility to combine this setup controlling the humidity and the temperature. For sure, we wanted to compare the nanostructure and the mechanical properties between two proton exchange membrane. This setup is developed between ForMAX beamline and MAXIV and the Chalmers University. We are using dynamical mechanical analysis combined with the simultaneous SWAXS that allows to prove storage and loss moduli as well underlying atomic and nanoscale structure into the material. And the experiment can be carried out in controlled temperature and humidity. That's a really complex setup. It's available now, so you can apply if you are interested. So the samples for this study were two. It's the Nafion membrane that is produced for DuPont company. This membrane is like really well studied and used. You can find thousands of paper in Google Scholar. This membrane is mainly used for fuel cells that use hydrogen as a fuel. And then also we work with the FumaPen F14100 membrane that is produced by another company, Fumatech. This membrane is less studied and less used, but it's more useful for membrane that use methanol as a fuel. Both membrane has like similar chemical structure. They have like a backbone that is a fluorocarbon structure, and then they have like a side chain that is finished in a sulfonic ionic group. This part of the molecule is hydrophilic and organized into the membrane in a way that they form water channel. Here I have a schematic. So where we have like a water channel in our cells, they are responsible for the conductivity process of the proton. Can be like a diffusion of the proton, Grotius effect, where the proton is jumping between the water molecules, or also can be like through surface conduction using the ionic group. Even if this two membrane has similar properties and similar structure, these water channels takes different shapes. In our previous study, we determined that Nafion membrane is formed by a cylindrical core shell water channel organized in hexagonal cluster. So here this light gray is the water channel, then the shell is the sulfonic chain, and then we have the matrix mainly for the fluorocarbon backbone in amorphous polymer. Also, we have like crystalline polymer, but it's not represented here. On the other hand, PEM membrane is organized in lamellar channel, also with the shell that is covered by the sulfonic groups. These two models were developed using Monte Carlo simulation, and also with a small angle scattering, neutron and X-ray. So it's another point to focus the powerful tool that is a small angle scattering techniques. So that is the setup that we use. Here we have the rheometer with the chamber that is able to control the temperature and the humidity. For this now are coming the X-ray. Here we have the window that goes through the chamber. This one here is the wax detector, and this for this little window, we can access to the SAT detector that is under a vacuum tube, like behind the wax detector. So this one are the parameters that we use, 20 kV for the energy, then the Q range that we can cover. This is the SWAXS range, and this is the PAX range. For the rheology experiment, we take a shared oscillatory test. This is the parameter that we use. And we control like a relativity humidity ramp going down and up from 5% to 55 at a temperature of 80 degrees, with the intention to simulate the operation condition of fuel cells. So this is a characteristic structure of a proton exchange membrane, not only from these two that we are studying, also a lot of another membrane has similar shape. We can find a ionomer peak that will be related with the water channel periodicity, how these channels are organized in cluster. Then here we have a matrix name that will be related with the crystalline domain that sometimes are formed by this fluorocarbon backbone in the chemical structure. Sometimes if we are further enough in a low Q range, we can get this power law that is related with the large inhomogeneities that we can find in the membranes. So now we will see how these three characteristics are related with the humidity and the structure in the membrane. So let's start with the ionomer peak, particularly the position. When we increase the relativity humidity, the position starts to increase in intensity, and also the center is moving to a lower Q values. That means that a correlation distance is start to increase in the sample. So that occurred for both samples. And here we plot 2 cycles going from 5 to 55% in humidity, this correlation distance. So we see that for NAFIO membrane, it starts to increase, like following a linear trend, but in FUMAPEN start quite constant, and then start to increase more slowly. This distance that we are representing here is for hexagonal cluster in the NAFIO membrane. This distance between the center of this two planar, and then for lamellar is the center between lamellar. So we are guessing that the way that the membranes are taking water is different between the membrane. Also, how the water is filling the channel sometimes take more time filling the FUMAPEN membranes before the channel start to increase in size. So if the channel increase in size, there will be an increase in the distance between the centers. Then if we analyze the area of our peak, we can relate if there are an increment in the amount of water channel available to produce the conductivity. So here we normalize the area of the peak. We can see the NAFION is increased like more than the FUMAPEN membrane. So, but both increase in size. That means that we have more cluster, more water channel, that implies more conductivity. If we compare the water uptake capacity of both membrane, we see that is quite similar in percentage, it's around 20%. But if we compare the conductivity of both membrane, like in this one is for environment humidity, is quite similar. But then if we go to fully hydrated membranes, the NAFION is like quite bigger than the FUMAPEN. So we can relate this increase in the area of the peak when the humidity increase. Then we analyze the matrix knee that, remember, is related with the crystal in our sample. For NAFIO membrane, also is an increase, and moving to the position of the PIP is going to a lower Q values. That implies that we have a bigger correlation distance in our sample. But for FUMAPEN membrane, it's like remains quite in the similar position. We can see in this plot that FUMAPEN membrane is like quite stable. The NAFION increase a bit. It's not a big difference between 5 and 55%. So we correlate this increment in the distance that is when the membrane is taking water, the water channels start to increase in size. So that makes the crystal start to, the distance between crystals start to increase. So we proceed to analyze what happened in the wax part, that, as you know, the wax profile gives more information about the crystallinity of our sample. This one is the typical profile in a NAFIO membrane for wax. We can decompose this peak in two. One, and this position that is related with the amorphous part, and this one at this position that is related with the crystallinity of our sample. These two positions we calculate in our previous work. I have not plotted here. So, but we can see that the amorphous peak is decreasing with the humidity, but there are not too much different in the crystal part. So we guess that this decrease in the humidity is because we have less contrast now with the amorphous part, because we have more water inside the membrane. So there are not too much scattering contrast between these two parts. And then for ForMAX membrane, we can see that at different humidities, there are no variation in the wax pattern. At the end, we analyze the power law that, remember that we can have this only for ForMAX membrane. Nafion membrane, probably it has a power law in the tail of the curve, but we cannot access because it was like at lower Q value. Here we have this parameter that I present at the beginning. That is the exponential parameter that decrease with the humidity, but it has value between 4 and 3. So that means that our channel has surface fractal. This parameter is related with the dimension. So the fractal dimension of our channel start to increase with the humidity. That means start to decrease the order in our molecules in our channel. So we are trying to find the connection, but for this study, we know that the water starts to bond in different way when the membrane is uptaking water. At the beginning, there are a layer of water molecules that are bonded with the sulfonic group. Then we have a second layer that is weakly bonded to the hydrophilic part. At the end, we will have free water. So these processes are modifying the surface of our channel that we can see here in the change in the fractal dimension in our sample. So for the mechanical test, we studied the storage module. Here we have the difference between the Nafion and ForMAX membrane. For Nafion membranes, both like a trend like linear decrease, but in the ForMAX it's more exponential way. The difference between these two ways can be related as the way the membrane absorbs water, that also we can see in the sample position, that we also see a linear trend in Nafion membrane and exponential trend in ForMAX. So we are developed what is the connection between these two graphs. The mechanical part is the water molecules act as a plasticizer, making the material more flexible. On the other hand, we are not pretty sure about this trend, so we are doing like extra experiment to have to be sure that that is a real mechanical behavior of our sample. And then at the end, like a conclusion or perspectives, is like we successfully implement this new setup that combine DMA with the SWAXS and humidity and temperature control at ForMAX. Is available for users. So if you are interested, you can write me and you can write a proposal. It's just for remake if you are working with a circular economy, or also for a normal way proposal at for MAXIV laboratory. We study and compare the nanostructure of the mechanical properties into proton exchange membrane under conditions like those operation. We relate the humidity with the membrane structure. And as a perspective and future work, we need to repeat a few DMA experiments. So thank you for